Hello everybody, I'm Hyung from Korea. I'll give a talk about the NGS study in neurological diseases. The contents will be as follows. First, introduction to medical NGS. The history of sequencing is very long. From the beginning, when Crick and Watson discovered the DNA structure, sequencing of the DNA was tried. As you can see, many genome assembly was founded. Sequencing tools also was developed. Sanger sequencing, which is still used frequently, is kind of a first generation. And NGS, next generation sequencing, and now third generation of sequencing method is used. NGS is applied in several fields. First, DNA sequencing using whole genome sequencing. Whole genome sequencing is a comprehensive method to analyze an entire genome. It can detect single nucleotide variants, insertions, deletions, copy number changes, and large structural variants. So it is ideal for discovery of variants and novel genome assembly. As you can see, human genome consists of gene and intergenic regions. Whole genome sequencing covers all of these genome area. When we do whole genome sequencing, making reference genome sequence is the first thing we have to do. Based on the reference genome, we can identify individual genome sequences. Usually, reference genome sequence is the work achieved by government or big academic consortium, and researchers use the reference genome for their individual research. Human Genome Project is the first international collaboratory research program, which designed to complete all the genes of human. Based on these accomplishments, we can initiate many of the researches on human genome. Reference genome is DNA blueprint used as a standard for comparison in basic research and clinical settings. The current version of reference genome is called Genome Reference Consortium Human Build 38. If you visit Genome Reference Consortium site, you can see or download the reference genome data. Second part of the DNA sequencing is whole exome sequencing. Exome is the genome that takes all the exons together. It consists of 1 or 2% of the whole genome. So if you are interested in the genes which cause proteins, it's better to perform whole genome sequencing. Whole genome sequencing process is similar to whole exome sequencing. The only difference is you have to capture whole exome using capture process. The other processes are similar to whole genome sequencing. Targeted sequencing is next generation sequencing, which elucidate some defined small number of genes. Due to high depth of targeted sequencing, it is commonly used in the clinic, but it has some limitation. When we do targeted sequencing, we usually make some multi-gene panels. The targeted sequencing has higher depth and wider coverage, so it is very suitable for clinical application, especially when you have good biomarker panels compared to whole genome or whole exome sequencing. Gene panel usually includes 10 to 1,000 biomarkers. This figure shows the brief comparison of whole genome sequencing and whole exome sequencing and target sequencing. RNA sequencing is method which examines the quantity and sequences of RNA in a sample using NGS method. It analyzes the transcriptome, so it shows the information about the genes. RNA-seq is one of the most commonly used NGS method. As you can see from the figures, RNA-seq experimental steps includes sample collection, isolation of RNA, and making cDNA, and the following steps are similar to a DNA sequencing method. These days, single-cell RNA sequencing is commonly used. You can take tissue and isolate and sequence individual cells. Then you will have RNA expression analysis of single cells. Spatial RNA-seq is another method derived from the single-cell RNA-seq, but it provides information on location. Few years ago, I started the NGS study. Whenever you are a beginner, you have to know some concept of NGS before starting your study. First, Sanger sequencing versus NGS. In principle, the concept of Sanger or NGS technologies are similar. This figure shows the steps of Sanger versus next generation sequencing. The most critical difference between Sanger sequencing and NGS is sequencing volume. Because Sanger sequencing can sequence one DNA fragment at a time, while NGS deals thousands or millions of fragments simultaneously. Since Sanger sequencing has the ability for longness and gold standards of accuracy, it is perfect for sequencing single genes or sequencing small amplicon targets at the lowest cost, or sequencing up to small samples at a time, like microbial identification or fragment analysis or the genotyping. And it is very important to be used for the confirmation of NGS result. I also use Sanger sequencing to confirm novel finding. NGS offers fast turnaround time, and it takes very short time to complete the study. So NGS is perfect for interrogating larger number of genes at a time and finding novel variants. Or you can sequence samples with low input amount of starting material. Sequencing microbial genomes for pathogen subtyping is also can be used using the NGS analysis. Variant analysis is one of the most important and most commonly used study method in clinical medicine. We can use three different formats: quantitative PCR, or Sanger sequencing, or NGS. Usually it is targeted sequencing. Microarray is another option we can use to study variant analysis, but now NGS replaces the position of microarray. These three methods has its own benefits and limitations. For qPCR, benefit includes high sensitivity, quick and simple workflow, and capital equipment because PCR machine is already found in most labs. But it has limitations, including only examine a small set of variants, virtually no discovery power, low variant resolution, low scalability. If you use targeted NGS, it has benefits including expanded discovery power, higher analytical sensitivity, greater resolution, and etc. So you can choose one of these methods for your study by comparing the benefits and limitations. Sanger sequencing and qPCR is good choice if you need to interrogate small region of the DNA on a limited number of samples. Otherwise, targeted NGS is shoot for your needs because it allows you to screen more samples and detect multiple variant types across targeted area. We can compare three DNA sequencing methods: whole genome sequencing, whole exome sequencing, and targeted sequencing. By comparing the benefits or limitations of this method, we can apply one of these methods for your study. Usually, targeted panel sequencing has higher accuracy, while whole genome sequencing has lower accuracy. Whole genome sequencing sequences whole genome, so it is very good for discovery purposes. But the sequencing depth is lower, and it covers everything. So it can identify all kinds of variants. Targeted sequencing just targets small specific regions, and the sequencing depth is very high, like 500 or 1000. It also can identify all kinds of variants, and as long as you know the targets well, it is the most cost-effective method. Since three different RNA sequencing methods has different limitations and benefits, depending on the gross features or location information or resolution, you can choose one of these methods: bulk sequencing, or single-cell RNA-seq, or spatial RNA-seq. Another thing you have to consider before you start NGS study is the method: short read versus long read sequencing. Then, what is read length? NGS read length refers to the number of base pairs sequenced from the DNA fragments. The predominant difference between long read and the short read approach is the significant increase in read length. In contrast to short-read sequencing, which comprises less than 300 base pair, long-read has capacity to sequence on average over 10 kilobases in one single read, thereby requiring less read to cover the same gene. Computer programs are used to assemble the fragments into contiguous sequences. Read mapping is essential part of the NGS analysis. Because the genome of most organisms is too long to be sequenced as one continuous string. Using NGS short-read sequencing, DNA is broken into short fragments that are amplified and then sequenced to produce reads. Usually, biomimetics techniques are used to piece together the reads like a jigsaw into continuous genomic sequences. So read mapping is the process to align the reads on a reference genome. This figure shows how to map the sequences. You will have a set of reads fragments, then you will align those reads into the reference genome. Long-read sequencing has some advantages over short-read sequences in mapping. Usually, the longer the read, the easier to map. You can see the difference between short reads and long reads. If you use short read sequencing, there can be more chances of missing sequences data. That means there will be some sequence gaps. If you use the long read sequencing, long read map usually produces and covers larger variants and providing comprehensive variant detection. In case of some repeated sequences, short-read sequencing can miss the number of repeated sequences. As you can see in this figure, there are 6 repeats. If you use short-read sequencing, your result will tell there are just 2 reads. But if you use long read, you can tell there are 6 reads. There are many benefits of long reads, but it still has many limitations. There are many limitations of current long read technologies. Another issue you have to consider when you do the NGS study is sequencing depth and coverage. Sequencing depth or read depth describes the number of times that a given nucleotide in the genome has been read in an experiment. Coverage describes a relation between sequence reads and references. This figure shows what is read depth and what is gene coverage very clearly. In conclusion, the higher depth it is, the better to use NGS analysis. Now we will go into the NGS study in urological diseases. NGS is accelerating neurogenomics research by revealing the mechanisms behind complex urological diseases. The interplay between heritable and non-heritable mutations, epigenetics, and other factors require NGS level analysis to increase our understanding. A wide variety of urogenetic diseases have been discovered owing to the development of NGS technology. NGS technology has led to great advances in understanding the causes of Mendelian or complex urological diseases. Owing to the complexity of genetic diseases, the genetic factors contributing to many rare and common urological diseases remain poorly understood. The clinical application of NGS significantly accelerates the discovery of disease-causing genes and promoted the understanding of molecular genetic mechanisms associated with hereditary diseases. Selecting the correct genetic test based on cost effectiveness, coverage area, and sequencing range can improve diagnosis, treatment, and prevention of neurological disorders. Now I will explain about some of my experience in rare unidentified neurological disease study. This study was initiated by enrollment of neurological disease patients in an outpatient clinic, and basic medical evaluation was performed, and the pedigree study was performed. Whole genome sequencing using blood cell was performed, targeting the patients and their family. And when we found out the new mutations, the confirmation was done using Sanger sequencing. The first case was a 6-year-old boy. He come to the clinic with the symptom of dystonia and congenital blepharoptosis. At first, the diagnosis was ophthalmological disorders or urology and neurodevelopmental disorders. But the clinician who take care of this boy suspected this disease will be some unknown urological diseases. So she interviewed with the family members and find out the mother has a mild symptom in the past, and the father was without symptom. During the whole genome sequencing, we identified likely pathogenic variant of SPAST gene. It was heterozygous, and this was observed in patient himself and his mother. We tried to do the further study, but because of the refusal of the patient, it was stopped. Another case was 23-year-old man. He come to the clinic with palmar facial fibromatosis. The initial diagnosis was suspected rheumatological disorders or different diseases. His father has similar symptoms, and mother has no symptoms. When we performed the whole genome sequencing, we find out some new likely pathogenic variant of DDR2 gene, heterozygous, in the patient and his father. These cases, we also could not perform the further study. Another case was dementia cohort study. This study was done in a prospective cohort. The people over 55 years old from the community was enrolled. First, a medical evaluation was performed, and neuropsychological and cognition test was done for all the people who are enrolled, and whole genome sequencing using blood cell was performed. Blood and CSF biomarker was evaluated. These biomarkers are the protein biomarkers, and image analysis using PET, MR, and functional MR was done. These two studies, the rare neurological diseases and the dementia cohort study, was performed for more than 5 years, but it was stopped because the funding was terminated last year. During this NGS study, I can expect some future issues. First, NGS study is very cost-consuming study, and also we need big resources. So cost and resource matters is one of the biggest obstacle to perform the NGS study in neurological diseases. These days, the wet lab cost is going down, and its speed is getting faster. So dry lab analysis becomes a bottleneck. In addition to the wet lab cost, so it is big issue to decide when do I start NGS study. Actually, during the study, I collected some patient samples, but some samples we didn't perform any NGS wet lab, and we still have many dry lab analysis to be performed. The second issue was integration of multi-omics data. At the beginning, we just planned the whole genome sequencing or whole exome sequencing, but we realized we needed to perform the transcriptome RNA-seq analysis, and also epigenomic studies are required in some cases, and we also have to add other omics like proteomics or metabolomics study. But we had some trouble in integrating these multi-omic data into a meaningful result. So we tried to develop the multi-omic integration and analysis pipeline, but it is just at the beginning stage now. Another issue was quality of the study. The quality of the sample for the NGS itself is very important issue. How to collect it and how to store the sample is very big issue. And in addition to the NGS data itself, we need to have plentiful of metadata. So how to access the metadata and the clinical data related with the patients or the normal paper is another big issue. During the analysis and the wet lab, both in wet lab and the dry lab, standardization of the protocol and quality control of the data analysis pipeline was also big trouble. The other issues, what I realized that recently is data sharing and disparity. We collected data for years, but we had a limitation in sharing those data because of some standard format and also accessibility, because our study was funded by the government and we don't have enough right to open those data. And also we had to get the permission from the patient or any people who provided the samples. And another issue was disparity, because the study which I performed is limited to Korean population. But when I want to compare the data with the other ethnicity, I recognized it has some trouble in direct comparison. And also, as you can see from many papers, the racial disparities or the basic disparities in designing the large cohort study has troubles. So I expect this data sharing and disparity will become a big trouble in the future. I briefly shared my experience in the NGES study in neurological disorders. Thank you for your attention. I really appreciate for me to have a good opportunity to present my experience in front of you. Thank you.